Hi, as I mentioned in the earlier class that I will be discussing the structure of DNA, DNA replication, transcription, translation, and how the genes are regulated. So, in the first step, the structure, I am showing the simplified structure of the DNA. This is the double helix DNA structure. And here you can see A, T, G, and C, adenine, guanine, cytosine, thymine, which are the basic components of DNA. So adenine binds with thymine with double hydrogen bond and guanine binds with cytosine with triple hydrogen bonds. As you can see here, these are the triple hydrogen bonds and then these are the double hydrogen bonds. So the double hydrogen bonds and triple hydrogen bonds can be clearly seen here with the dashed line. And this is the same representation for the ATGC bindings. And then this is also basically showing the replication part, which I will be discussing in the next slides. Now, what is central dogma? Where I am starting. So central dogma is basically the formation of RNA and protein. So basically stepwise you can see DNA to RNA and then RNA to protein, DNA to DNA, RNA to then DNA to RNA, then RNA to protein. So DNA to DNA is replication, and then DNA to RNA is basically transcription, then RNA to protein, that is translation. So the replication you can see here clearly, this is the self arrow, and then this is transcription, blue arrow, and the red arrow is showing the reverse transcription, that means RNA to DNA. And then RNA to protein, here you can see with black arrow, that is translation. So the central dogma of molecular biology, which was first proposed by Francis Crick, describes the one-way genetic information flow. DNA to RNA and RNA to protein. It cannot be reversed from protein to RNA or RNA to DNA. So this is unidirectional process. DNA, the genetic blueprint, is first transcribed into messenger RNA or mRNA, which then carries the information or instructions to the ribosomes, where it is translated into proteins, that is the functional molecules that carry out most of the cellular tasks. Now, the same thing, the central dogmas can be presented in different way of pictorial representations. So this is here you can see DNA to DNA, that is replication, and then DNA to RNA, that is transcription, and then RNA to protein, that is protein translation, and then this is showing the protein, that is phenotype or functions you can see. The same thing you can see here, DNA replication, DNA to DNA, and then DNA trans to RNA, that is here transcription, and then RNA to protein, that is translation. And the same thing can be seen here. So next part is what you could learn from the breakdown of the process. So DNA, the molecule that stores the genetic information or code in the nucleus of the cells. Transcription, the process where a DNA segment is copied to create messenger RNA molecule, that is mRNA molecule. RNA, a temporary copy of DNA's genetic information acting as a messenger. And translation, the process where the information in the mRNA is read and used by the ribosomes to synthesize a specific sequence of amino acids forming a protein. And protein, the final functional product that performs various roles in the cells. And here the same thing you can see, DNA to RNA, that is transcription, RNA to protein, that is translation here. So here this is representing the double-stranded DNA. This is mRNA and single-stranded RNA. And then here amino acid sequences, that is for protein. Now, key aspects of the central dogma. Information flow, genetic information primarily flows in one direction and from nucleic acid to protein. Unidirectional information cannot be transferred back from protein to nucleic acids. And replication, DNA can be replicated to create more copies of itself, maintaining the genetic information. Expanded central dogma. The central dogma was later expanded to include the processes like reverse transcription, where RNA is used to create a process important in some of the viruses. And the same thing here you can see the DNA, double-stranded DNA, annotated with ATGC, and then the bonds also. RNA here, these are the ATGC bases, and then RNA. After RNA, there is protein sequence, amino acid sequence for the proteins. And next part is what is DNA replications. So in the sense that DNA replication must occur if genetic material is to be provided for the progeny of any cell, whether somatic or reproductive. The copying from DNA to DNA arguably is the fundamental step in the information transfer process. A complex group of proteins called the replisome performs the replication of the information from parent strand to the complementary daughter strand. And the summarized form of DNA replication, here you can see the 5 steps. For example, cases unwind the parental double helix, and this is the enzyme, and this is the double helix, parental double helix. And this is, you can see here, 5 prime to 3 prime and 3 prime to 5 prime, 2 strands. And then here you can see second step is single strand binding. Proteins stabilizes the unwound parental DNA. And afterward, this third step is the leading strand is synthesized continuously in the 5 prime to 3 prime direction by DNA polymerase. And then fourth step is the lagging strand is synthesized discontinuum. And then RNA primer, which is extended by DNA polymerase to form the Okazaki fragments. And then finally, the fifth step is after the RNA primer is replaced by DNA, that is here, this DNA ligase joins the Okazaki fragments to the growing strand. And then you can see here some of the terms. DNA ligase, what is the purpose of ligase? Just to connect with each other, all the fragments. And then what is here? RNA primer, and then replication fork, then DNA polymerase, and then Okazaki fragments, and DNA polymerase, primase. All these terms and terminology you need to remember. And then next part is here you can see the same DNA replication process as a summarized graphical presentation. So lagging strands, you need to understand what is lagging strand, what are topoisomerases, parent DNA, original DNA, primase, helicase, RNA primer, Okazaki fragments. And then you are fully familiar or introduced with DNA replication. Now it comes transcription process. Transcription, as related to the genomics, is the process of making an RNA copy of the gene's DNA sequence. This copy is called messenger RNA or mRNA, which carries the gene's information, protein information encoded in DNA. And here you can see, and the location of this DNA is here in the nucleus. This is just to show the location of the material. Here DNA, again it is at nucleus level. Then mRNA is finally moving out of the nucleus and going to the cytoplasm for the protein synthesis or translational process. So you can understand how the copies, replication copies DNA for cell division. Then how this mRNA transcription process takes place too, which makes mRNA from the DNA. And finally, when you have RNA, then translational process completes the protein synthesis part, but with the help of different organelles or processes. Then you can see in the representation here this nucleus, from nucleus how it goes out to form the protein by passing through the ribosome. And then this is the final step. So you can understand how the transcription, translation, and replication could take place, and what is the location of transcription, translation, and replication process within the cell. Now, DNA replication copies the entire genome to pass to daughter cells, resulting in two identical DNA molecules using DNA polymerase and AT base pairing. Transcription creates an RNA copy of a specific gene for the protein synthesis, producing a single stranded RNA molecule using RNA polymerase and an AU, that is instead of AT pairing. The key distinction lies in the their purpose, product, template use, enzyme involved, and the base that pair. So you need to remember what is the difference between replication and transcription. So here it is clearly mentioned that the key distinction lies in their purpose, product, template use, and enzymes involved, and the base that pair. That the same thing you can see here. Now, the purpose, difference at purpose level. To create an exact copy in replication, you create an exact copy of the entire DNA genome before cell division. While in transcription, to copy the genetic information from a specific gene into an RNA molecule, so guide protein synthesis. In terms of product, in replication, two identical double-stranded DNA molecules, while in terms of product, a single-stranded RNA molecule is formed. In case of replication, the template, both DNA strands act as templates, while in transcription, only one DNA strand acts as the template. In the enzymes used in DNA replication, catalyzed primarily by DNA polymerase, while here catalyzed by RNA polymerase. Then base pairing is somehow slightly different. Adenine pairs with thymine and cytosine pairs with guanine. While in case of transcription, adenine pairs with uracil, while cytosine pairs with guanine. So the difference is pairing adenine pairs with uracil. Now, this is basically if somebody is trying to understand what is transcriptomics and what do we study in transcriptomics. In transcriptomics, we mainly study mRNA, mRNA, rRNA, transfer RNA, tRNA, long non-coding RNAs. Now, transcription is the process of copying a segment of DNA into RNA for the purpose of gene expression. Some segments of DNA are transcribed into RNA molecules that can encode proteins, that is called messenger RNA or mRNA. Other segments of DNA are transcribed into RNA molecules called non-coding RNAs, that is ncRNAs. Both DNA and RNA are nucleic acids composed of nucleotide sequences. During transcription, a DNA sequence is read by an RNA polymerase, which produces a complementary RNA strand called primary transcript. In virology, the term transcription is used when referring to mRNA synthesis from a viral RNA molecule. The genome of many RNA viruses is composed of negative sense RNA, which acts as template for positive sense viral messenger RNA. A necessary step in the synthesis of viral proteins needed for viral replication. This process is catalyzed by a viral RNA-dependent RNA polymerase. And this thing, you can see here clearly how the normal transcription looks like and how the viral transcription could look like. So the left side figure is normal transcription process, while the right side figure is for viral transcription. Now, a DNA transcription unit encoded for a protein may contain both a coding sequence, which will be translated into a protein, and the regulatory sequences, which directs and regulate synthesis of that protein. The regulatory sequence before in the coding sequence is called the 5' untranslated regions. So that is 5' UTR. And the sequence after downstream from the coding sequence is called as the 3' untranslated regions or 3' UTRs. As opposed to DNA replication, transcription results in an RNA complement that includes the nucleotide uracil in all the instances where thymine would have occurred in a DNA complement. So this is the transcription unit in the figure which you can see here, 5 prime to 3 prime and 3 prime to 5 prime. So template strand, a structural gene and coding strand. Then you can see promoter and the terminator. Now, only one of the two DNA strands serves as a template for transcription. The antisense strand of DNA is read by RNA polymerase from 3 prime to the 5 prime end during transcription. The complementary RNA is created in the opposite direction, in the 5 prime to 3 prime direction, matching the sequence of the sense strand except switching uracil for thymine. This directionality is because RNA polymerase can only add nucleotides to the 3 prime end of the growing mRNA chain. This use of the 3 prime to 5 prime DNA strand eliminates the need for the Okazaki fragments that are seen in the DNA replication. This only removes the need for an RNA primer to initiate RNA synthesis, as is the case of in DNA replication. And here you can see the DNA 5 prime to 3 prime, 3 prime to 5 prime, promoter, RNA coding sequence, and terminator. So here transcription initiation site in the promoter site, and then here in the termination site, transcription termination site. So upstream of the gene is left side, downstream of the gene is right side. So now you can see the non-trans template or sense nonsense strand of DNA is called the coding strand because of its sequence is the same as the newly created RNA transcript. This is the strand that is used by convention, which when presenting a DNA sequence. Transcription has some proofreading mechanism, but they are fewer and less effective than the controls for copying DNA. As a result, transcription has a lower copying fidelity than DNA replication. Transcription is divided into initiation, promoter escape, elongation, and termination. So regulation of transcription in mammals can be seen in the picture here, and this illustration indicates many of the elements that are present when transcription of a gene is upregulated. Now, in the next part, I will be discussing the translational process, where we will be continuing how the mRNA to protein is synthesized, and that is translation. So, which I will be discussing in the next class. So, thank you very much, and please subscribe to keep updated about the next tutorials. Thank you very much.